Hi friends. See, in this lesson, in this session, we will study about three types of simple permanent tissues. So the division we already discussed, isn't it? Yes. So, which are the three simple permanent tissues? What are they? We know very well. They are parenchyma, collenchyma, and sclerenchyma. So, at a stretch, we can study all these three by using certain tips. See, first we will think about the shape of these three tissues. By comparing, we can easily study. See, shape. In case of shape, parenchyma cells may be round, oval, polygonal, or elongated. So parenchyma cells may be round in shape, oval, polygonal, or elongated. But at the same time, the collenchyma cells are seen round, oval, or polygonal. Okay. See, parenchyma cells can be seen round, oval, polygonal, or elongated. But collenchyma cells are mostly found round, oval, or polygonal. And in case of sclerenchyma, they are narrow cells and elongated cells. Narrow and elongated cells. So this is about the shape. Okay, the sclerenchyma cells are always seen narrow and elongated. Their lumen, inner space, will be very less as compared with the other two. Then life, about the life. See, parenchyma cells are living. So in parenchyma tissue, the cells are living. And here in collenchyma also, the cells are living. So both are having living cells. But in case of sclerenchyma, we can see the cells are dead. So cells are dead in case of sclerenchyma. Then coming to the intercellular space. See, in parenchyma there will be intercellular space. So cells are loosely arranged so that there will be intercellular space. So cells are loosely arranged, loosely packed with intercellular space. With intercellular space. So here in parenchyma we can see intercellular space. But in case of collenchyma, very little intercellular space is present. Very little intercellular space. So that is one of the peculiarity. In case of collenchyma, we can see very little intercellular space, but in case of parenchyma, intercellular space will be present because the cells are arranged so loosely with the intercellular space. But in case of sclerenchyma, there is no intercellular space. No intercellular space. That's it. So the sclerenchyma cells are actually dead cells. They are arranged so closely without any intercellular space. And another case, if you are thinking about the thickening of the cell wall, thickening of the cell wall. Here, the cell wall is made of cellulose. So cell wall is made of cellulose without any thickening. Without thickening. So in case of parenchyma, the cell walls are not having any thickening, and it is normally, as usual in plant cells, it is made of cellulose. But in case of collenchyma, if we check the collenchyma cells, the cell wall is made of cellulose, but the corners of the cell wall is thickened with a substance called pectin. So a substance called pectin. With that substance, the cell wall, the corners are thickened. So the corners of the cell are thickened with the pectin in case of collenchyma. And next, in case of sclerenchyma, around the cell wall, it is thickened. With lignin, a substance called lignin. So here, in case of sclerenchyma, we can see lignin is the thickening substance, and in case of collenchyma, pectin is used for thickening. But the thickening will not be all around, but it will be thickened only at the corners of the cell. And in case of parenchyma, no thickening at all. Then coming to the functions. So here, the parenchyma cells are used for storage, secretion, and for photosynthesis. So some of the parenchyma cells are having chlorophyll in it. If chlorophyll is present, such parenchyma cells are called collenchyma. So they can conduct photosynthesis. So, and along with that, secretion is done and also storage. So storage, secretion and photosynthesis are done by parenchyma cells. And in case of collenchyma cells, they are giving mechanical support to stem, petiole, etc. Along with this, they are also having chlorophyll in it. Most of the collenchyma cells are also having chlorophyll, and as a result, we can say photosynthesis can also be done. They can do photosynthesis also. And in sclerenchyma, they are giving mechanical support to organs. See the shells, see coconut shell, that shell is made of, coconut shell is made of the pod of different nuts. So all these are actually made of sclerenchyma cells. They are dead cells and they have a thickening with the lignin. So they are giving mechanical support to different organs of the plant. Okay, now we can check the figure also. How can we show the figure? See, the cells here in parenchyma are arranged with the intercellular space. So it is very easy to draw. So these spaces are called the intercellular space. So these are the parenchyma cells. They are living cells. Parenchyma cells are, so these are intercellular space. So here in parenchyma we can see intercellular space. Okay, but in case of collenchyma, intercellular space is somewhat less, and the cell wall at the corners of the cell wall are thickened with a substance we have said pectin. So we can draw like this. So the corners of the cells are thickened. So these are the corners. So here there will be a thickening, and this thickening is made of a substance called a pectin. So in this way we can draw collenchyma. So the corners of this collenchyma are thickened with a substance called a pectin. Then, so this is pectin. Here is cell wall is present, and cell wall is made of cellulose itself, and the pectin is present only at the corners in order to and sometimes it will be made of hemicellulose also. Hemicellulose. So either pectin or hemicellulose. This may be made of hemicellulose also. And in case of sclerenchyma, cells are elongated. They are closely packed. Cells are elongated, closely packed, and around the cell wall there is thickening. So thickening is present around the cell wall. So this is the thickening. So not only in the corners, but all around the cell wall we can see the thickening. And here and there we can see some pits also. This is the pit. So the pit means there is no thickening at all. So certain pore-like regions without a thickening. But there will be cellulose made cell wall will be present, but the thickening will not be present. So this region is this without a thickening. So these are some regions, they are called pits. It's a pore-like a region. So the thickening will be all around the cell wall. Around the cell wall there will be, and this inner region is called a lumen. The inner space of the cell in sclerenchyma is called as the lumen. And so this is the structure of parenchyma, collenchyma and sclerenchyma. From this we can describe about each one and the comparison. All this we can do. Okay, thank you.